Well, after 66 years working with mushrooms, I feel kind of drugged. But it's not because I've been smoking them or eating the wrong ones, but they're all around us. We'll talk about some of those things tonight. Seasonally, I'll be talking about this particular mushroom a bit tonight in terms of its genetics and the fact that there are 7 different subspecies, if you will, of this particular mushroom. I'm sure you all recognize it. But only in Alaska are all 7 species found in this continent. And they're found throughout the world, but only in Alaska. So we're kind of a hotspot for these things for sure. My research has focused on circumpolar events, both north and south. So I'm kind of bipolar in my wording and my actions, but starting with Alaska, the coastal Arctic slope, the interior of Alaska, northwest Alaska, which is really mostly Arctic, the southwest Alaska, which includes the Aleutian chain, south central Alaska, and southeastern. It's huge. It's too huge to give a simple presentation on mushrooms of Alaska, because every one of these ecosystems has an entirely, not different, but a unique component of its mushroom flora. Some of the things that you might see if you went to these places, like Barrow, Alaska, now called Utqiagvik. In fact, there are 2 people here who met in Barrow, Alaska, and got married, and that is my brother-in-law and sister-in-law, Roland and Gretchen Durkin, which are sitting right here. If one travels south from the northernmost tip of land on North America's continent, you would pass from the Brooks Mountain Range into the Foothills Province and then into the Coastal Plains Province, which is really marked by a whole series of unique lakes that are northwest, southeast directed. And the reason for that is because the winds blow predominantly out of the northeast, east, and out of the southwest. And so when the winds hit the melting ice sheet on these ponds, it sets up an eddy effect, and that eddy effect moves up and grinds away at either the northwest or the southeast portion of the land, and the lake literally moves a meter a year one way or the other. So this tundra is alive, it's moving. Everything is constantly moving around. And you can see from this particular photograph, there's a section here that if you got right down on the ground, you'd find very large polygons, which we call hydri polygons. And sitting with William Campbell Steer, who was a great bryologist, studied mosses out of the New York Botanic Garden for years. He and I went on a field trip, and we had lunch sitting on one of these polygons, and he said, "Gary," he said, "you're sitting at 5,000 feet if you were in the Cascade Mountains." I said, "How do you know that?" He says, "Look at this moss." And he picked up a moss. He said, "You'll only find this at 5,000 to 7,000 feet in the Cascade Mountains." Then he said, "Look at that trough. That trough is 5,000 feet below at sea level at mid-latitudes. So the mosses we see down here are something you'd see at sea level in Washington, Oregon State." So it was just a realization to me that so much was going on in this place with all of these different things. And if you move west, in the Arctic and Northwest, you come across a town called Nome. You've all heard of Nome, I'm sure. It's a beautiful little city. There's a good deal of mountainous area to the east of it, and the tundra itself looks like a garden of lichens, grasses, and other vascular plants. It's a wonderful place. If one travels to the interior of our state, you'll see beautiful pictures like this of our great Mount Denali or Mount McKinley. Matter of fact, our home, maybe you remember this, our home, we had a chair we called the Denali chair. And 125 miles away, you could see Mount Denali from our great room, our living room. It was just awesome. But if you move around the interior a bit and you start moving back north, you're apt to see a landscape that looks something like this in the fall and like this in the early spring. And if you look at the components, you can see there's dark patches here, which are all black spruce trees, and they're all underlain by permafrost. Here, you have deciduous forest trees in the birch and the aspen, and they are not over permafrost ground. So we have discontinuous permafrost in the interior of Alaska, but you go north across the Brooks Mountains, and it's continuous permafrost throughout the land. And if you go down into the south central region, our banana belt, we find some beautiful lakes and beautiful trees. We get into not only the black spruce, some areas white spruce, and Sitka spruce and larch, but we also have the places like Palmer, where I'll be going and teaching again this summer, beautiful mountain backdrops and farmlands that have been farmed now for many, many years, and it's the breadbasket of our Alaskan state. We also find that our tundras or surfaces are lichen rich, and lichens are in fact fungi. In fact, they are cup fungi for the most part, and we have an expert in our midst. And this young man, Tim Wheeler, has studied the lichens extensively and has a wonderful collection. And we'll be talking on 9 April about fungi that are edible, right? And so Tim, I introduced him as also a graduate student, the first PhD graduate student in mycology at UM. So welcome, Tim. And so we also get out into the Aleutian Islands, beautiful place. Dutch Harbor is on its own little island, and there's a bridge that connects Dutch Harbor to Unalaska Island. And if you go further interior, you come across braided streams, all kinds of landforms that really shape what we know to be interior Alaska. And that doggone river is 1,982 miles long. And it's a wonderful river, a main thoroughfare for lots of folks up and into. One of the things that really interested Tim and myself recently is this island called Casatoche Island, and it blew up. It was a beautiful island in the Aleutian chain, about halfway out, just north of Adak Island, northwest of Adak Island. And in 2008, the whole island, the volcano blew up, and it destroyed every living thing on the island. But living things are starting to come back, as they always do. But the first things that came back were mushrooms. An orange peel mushroom I'll show you later, and a couple that Tim and I are working on in the genus Foliota, and I'll show pictures of those later too. And they're all considered to be fire-loving fungi. As you can well imagine, that hot ash must have been a tremendous scarring effect down as it poured down the slopes of that mountainous island, and small island too, by the way. But some of those fungi that we associate with cataclysmic fire events are given several names. I've often just referred to them as pyrophilous or fire-loving fungi, but terms like phenacoid, carboniculus, and anthracophilus are all terms that mean the same thing. Mycologists love terms. It's amazing. And so we see things like Geopixus carbonaria, this little fella. It's a stalked cup mushroom, and the stalk is about 2 to 3 times as long as the cup is wide, but it's buried in the substrate. You can't see that. Besides the violaceus you see here, Plicaria carbonaria, see there's the name carbonaria, right here growing on burnt ground. We have Pholiota brunescens, something that Tim and I are working on. Pholiota castanea, Pholiota highlandensis, another. These two were the first gilled mushrooms to show up on that island that I showed you. And then we have Pulvinula archii, another cup fungus, and a Rhizenia undulata, which is a real interesting mushroom. We do have it in Alaska, but rare. In fact, it's more commonly seen in fire-burned areas in the East Coast, whereas the mushroom we predominantly see in fire burns in the western coastal areas is what? The morels. The morels. They both are cup fungi, but they're totally different species for East and West Coast in fire-burned regions. And then we've got, and that is that Rhizenia, and then we've got Tarzetta cupularis right here, tiny little guy forming a beautiful cup, and hence the name cup fungi. Well, if we look at Southeast Alaska, we are truly in a cold, temperate rainforest, and mushrooms abound. And you can see not only the thick undergrowth, but the very thin trees that are just striving to get a little bit of that sunshine in that canopy. And of course, as you have probably seen or perhaps even traveled to, we have glaciers all over the place. And this is the chunk of glacial ice right off the coast where we're collecting. And you can see these little white patches that follow the breaking zones of the ice. And those little bubbles are filled oftentimes with fungal spores. So we have determined from ice that is millennia old what some of the fungi must have been at the time that that ice was formed. But another thing that I'm going to try to do is dispel some of the toadstool mushroom fallacies, if you will. Some of the wives' tales. And somebody mentioned, Tia mentioned in our first, that mushroom that was found in the swamps of where, Chicken? India, had mushroom growing on it. And I'll come back to that. But it's the first truly fungus that I have seen growing, a basidiomycete growing on an animal like that. So people have asked me, well, Gary, how many mushrooms are there in Alaska? Everyone thinks it's rather cold, dark, and dingy, and not much grows there. But we have found in working in our sub-Antarctic and Arctic, circumpolar Arctic, circumpolar subarctic, and circumpolar sub-Antarctic island archipelago. In other words, we do not work in Antarctica, but we work in the islands between the continents in the Southern Ocean. And we have found that there are about 12 species of fungi for every plant species. And so with a little bit of math, we can see that Alaska provisional plant species, about 2,773 species of plants in Alaska. There are approximately 1,388 species of mosses. And for high latitudes, the 12 fungi in these two groups, we would see typically 49,932 species of fungi in Alaska. But if you consider the small, very inconspicuous fungi in the Ascomycota, the cup fungi, that make up about 75% of all fungi, that we should see somewhere between 12,400 and 12,500 species of gilled mushrooms. Have we seen all of those? No way. It's just too big a state to get to them all, but we've seen quite a number of them. You were asked the question, why are fungi not plants? They have no vascular tissue. But how do they get stuff from the bottom of the mushroom up to the top of the mushroom? They have tubes that conduct stuff up and down. It's just not vascular tissue, as you see in woody plants. And that's why woody plants grow tall and mushrooms don't grow very tall, except there is a mushroom in Africa that grows on elephant dung. It's about this high. That's a big mushroom. They've got no chlorophyll, but yet some of the mushrooms are green, so they can't produce their own food. They need sugar. Oh, they're sugar lovers. But they do not produce their own sugar or glucose like plants do, and upon which you and I are very dependent. They don't have seeds. They have spores, spores being much tinier than seeds. No fruits, but they have what we call a fruit body. So when you pick up a mushroom, you're handling the fruit body. No true roots, no true stems, no true leaves. Instead, they have rhizomorphs, which are root-like structures, stipes, which are stalk-like structures or stem-like structures, and polii, the caps, which essentially are kind of leaf-like in some of their function. So what do they do? How do they do it? We ask these questions oftentimes. So mystical, magical mushrooms, what's really going on, I think fascinates a lot of folks, particularly since now we're finding that some of the hallucinogenic fungi are producing biochemicals that are going to be very, very useful medicinally for treating PS, some of our folks coming back from war zones and whatnot. But all mushrooms are fungi. Some are called toadstools, but a mycologist does not discern between a mushroom and a toadstool. To me, everything is a fungus or a fungi. If you're British, fungi. Where do they grow? Everywhere. They'll even grow on the lenses of your camera, believe it or not. How do they grow? I'll tell you what. For the most part, quite quickly, but they can be rather slow and melodical, particularly in some of the fungi that grow on trees we call bracket or shelf fungi, may last for years. I mean, they may grow for 10, 12, 15 years and just get bigger and bigger and bigger with time. How do they eat? They don't eat like animals eat, but they do release enzymes from their hyphae or their very small tubular cells, and they exude those enzymes to the outside of their cellular body. Those enzymes digest the material extramural to the fungus, and then the fungus literally grows into and around and sucks that stuff up by absorption. So they're absorbers. So you've seen a green mushroom? There's some good edible green mushrooms. And we find that many of these mushrooms fall into one of four different categories. They're either saprophytes, which means they live on dead stuff, and they are great decomposers. In fact, I suspect, and I'm guessing that probably 52 to 54% of the mushrooms that we find right here are decomposer mushrooms. And the others, for the most part, the higher fungi, are going to be mycorrhizal species. And I'll talk about that in a minute. But if they grow on mosses, we call them bryochilous. If they grow in fire burn areas or around campfire pits or whatnot, we call them carboniculus or pyrophilus. Now you've got three other terms you can use. If they are dung-loving, oh, mushrooms love dung. They're called coprophilous. If they grow on insects, they're insectivorous. If they grow on skin, feather, and nails, oh, they love you. We call them cratinophilous fungi, like toenail fungi, fingernail fungi. If they're growing on lichens, we call them lichenicolous fungi. And a very dear friend of mine from the Komarov Botanical Institute in Russia, St. Petersburg, is a specialist in this area and actually named one of those little creatures after me. I'll show it to you. It's called Polycoccus larsenii. And you'd never recognize it if you saw it, I'm sure. I don't even know if I would recognize it if I saw it. They're so small. If it's growing on dead wood or dying wood or living trees, which the wood is actually non-living, they're called lignicolous. If they grow on rocks or glass, siliceous, and of course, many on the ground, terra firma, they're turriculous or geophilous. If they grow underground, they're called hypogeous. If they grow above ground, they're epigeous. So we find them everywhere. So we've looked at the forests of the interior of Alaska and defined some of the cycles of our forests. And we can start with a good old healthy tree. This is actually a black spruce tree. However, the limbs are sticking straight out or slightly up that would suggest a white spruce. But in its root system, in the rhizosphere, there are a number of fungi that are growing. And all these little black dots are mushrooms that are growing underground and are associated with the roots of that tree, without which that tree would not be living there. Then there are epigeous mushrooms right here, fruiting above ground, but still associated with the roots of that tree. Then along come small mammals, flying squirrels and the red squirrel and the red-backed voles in our country that eat these mushrooms. And they go prancing around, and they oftentimes take those mushroom fruit bodies and put them up in the limbs of trees to dry them out. And they store them. I have seen vast storages of mushrooms in people's outbuildings in their attics. The whole attic might be filled with dry mushrooms. It's just amazing. And they do go after those that are hallucinogenic. And I suspect, because squirrels are kind of active pretty much year round, that if you see a squirrel in the wintertime and it's kind of yelling or screaming at you, he's probably high. But then we find mushrooms like the rusts that grow on the needles of the tree. And where these rusts form, you get a tangle of limb branching because of the hormone released by the fungus to stimulate branching in the tree. We call that branching pattern a witch's broom. And the witch's broom is a place where our small creatures will go into and build nests, raise their kids, and once their kids are out of the nest, they fill those nests with dried mushrooms. So they use them as caches, fungal caches, and they eat mushrooms all winter long. Well, eventually, the tree will succumb to other forms of fungi and will fall to the forest floor and begin its decomposition process by a whole host of other fungi. But these logs are used by our squirrels as runways through the forest. And when they defecate, the excrement loaded with spores roll off these logs, and generally on the downhill side, those fecal pellets are just spore bombs, just waiting for spring to come on, a little bit of moisture, and boom, all those spores start germinating, and you get fungi all over the place, but particularly along the downhill edges of those logs. And so we have a cycle that occurs in many of the between many of the organisms in the forest, and some of the fungi that we see in terms of the bryoculus mushrooms, those that grow on mosses, are seen here. This is actually a little chanterelle, Cantharellus umbenatus. And we also have many of the Gallorhinus species, too numerous to go to a species level here. So I just called it Gallorhina. We also have, growing on dead wood, those things that look like bones when they're actually sporulated. Otherwise, they'd be pure black. And these are lignicolous, actually growing on wood, not growing on moss. But the log was once completely overlain by moss. So one would think, well, this mushroom was growing on the moss, and it really isn't. It's growing on the wood. But this Spathulary flavida, the tongue fungi, also is a moss lover, as is another Gallorhinus species. So we have those things that are living on mosses. We have the coprophilic fungi, like Stropharia semiglobata, on horse dung. And I've seen these about 12 to 14 inches in height. So it's amazing heights that can be attained by some of these fungi. And then we've got small fungi that actually have a blasting mechanism that's hydrologically used to blast out its package of spores. And it's said that the speed of the blasting of that spore package out is faster than a speeding bullet. It's amazing the power that the fungus has to forcibly eject and shove that spore package out in Pilobolus. And they're dung lovers big time. Then we've got the Pineali. Many of the Pineali species are hallucinogenic, or at least have some of the hallucinogenic compounds within. But again, they grow on dung. And I've seen people climb over fences into people's farmyards looking for cow manure and dung-loving fungi. And people have come out with shotguns and say, "What are you doing to my land?" Point the gun and say, "Well, we're just here looking at species of fungi." "Oh, get off my land," you know. So you have to be kind of careful what you're going to be doing. But they tend to grow on moose pellets, and they grow on things like elephant dung. This is a big pile of elephant dung. Of course, we don't have elephants. Well, no, we do have an elephant in Alaska at the zoo. But I've never checked its dung for mushrooms. Whoa. And then we've got things that you and I eat that we buy in the store that are referred to as Agaricus campestris or Agaricus campestris variety bispores or Agaricus bispores or Agaricus brunescens. There's just a whole complex of things here. But these mushrooms are grown on dung, on manure mixed with straw in mushroom beds. And so when the mushrooms pop up, the folks that go in snip them above. And this is one of the reasons you never want to get these mushrooms wet by washing them. You want to take a little brush and brush the debris off and then cook them well, because there can be various ascomycete fungi and infecting bacteria on these things as well. And oftentimes if a person gets sick, the mushroom gets blamed, but it isn't often the mushroom. However, this particular mushroom also produces a substance that essentially is like jet fuel. It's called MMH, monomethylhydrazines. They're essentially a fuel-like component. But you'd have to eat a truckload of these things constantly to be contaminated by them. Just cook your mushrooms. And then we've got these little guys in microclivia that grow on dung as well. And until you pick the mushroom and look at the base of the mushroom, there's a little seed-like structure, which is the tuber that gives rise to the mushroom. And that tuber is the structure that's actually living inside the decomposing material, primarily mushrooms, old mushrooms. And then we come across all these things that we call lignicolous, or wood inhabiting things. And there's just a ton of them. These are all wood inhabiting. I must admit, this is a slime mold. And the slime molds, and this is another slime mold. There's another slime mold. And so these slime molds used to be in the fungal kingdom. But then they looked at what happens when the spores germinate, and they produce an amoeba. An amoeba moves by pseudopodia and contracts and moves out here and it crawls along and is a bacterial eater. If it's really wet, they produce spores that actually have little swimming tails called flagella. Nothing moves in the fungal world. And so you can guess that the protozoologists say, you mycologists are all wet. Those don't belong to you, they belong to us. Because they are sarcodinian, amoeboid-like things or mastigophorine-tailed things, and they are not fungi. And they ripped them out of our hearts and our souls and took them away. Miserable. However, once we got busy with DNA and started looking at their DNA composition, some of the water molds, which were taken away from us, were so close to the other fungi that mycologists were able to bring some of the water molds back into the fungal world. So in fact, we do have mushrooms that do have little swimming tails on some of their spores when they mature and grow. And of course, these are all wood-rotting fungi, and you'll notice them as bracket fungi, some of which are very edible, some of which you can actually dry and take an ember from your forest fire or your campfire, put it into the hole of the mushroom, dried mushroom, and it'll begin to smoke. It's a great insect deterrent. So when we're out in the woods, we'll take one of these species of wood-rotting fungi and dry it a bit, and then ignite the interior portion of it, and it smokes and it keeps away the mosquitoes really nicely. Some of them are teeth fungi that grow on wood. Others are gilled or pored mushrooms, but there is just a waft of different things that will grow on wood. And some of them, like this guy right here, Pitpora speculinus, is actually dried by native peoples, ground up and used and mixed with pipe smoking tobacco. And it's not hallucinogenic, it's analgesic. So it cuts pain. So it's like aspirin. Amazing. Then we have things like the lichens that Tim knows so well, that grow on wood, rocks, and other debris. Here, some of our siliceous lichens, crustose forms for the most part, are growing on rock substrates. You can see the rock here and here, but beautiful lichens that cover these wonderful sites. Then we've got those fungi that grow underground, or hypogeous fungi. They're terriculous in the sense that they grow in the soil, but they are underground. And boy, do the squirrels love them. In fact, they'll give off an acetylene-like odor that is hardly detectable by you and I, but boy, the little rodents in the forest can pick up those odors like you can't believe. And they'll go and they'll dig a hole the exact size of the mushroom fruit body and pull it right up and out and eat them. This Geoffra cooperi is actually known to be a delicious edible for us, but I've only found it growing in one place: old outhouse pits. And therefore, I just stay away. Some of them are rather beautiful. And of course, they all, in this case, have forcible spore discharge. But they're abundant. And we have identified about 17 species of hypogeous fungi of one group, the false truffles, and about 9 species of the true truffles. Yes, we have true truffles in Alaska. And here are some of the false truffles. And the reason they're referred to as being false is because they belong to and are associated with the actual mushrooms that have gills or pores. True truffles belong to those cup fungi groups that have spores that are forcibly discharged, yes, but their spores are borne in long tubular-shaped cells called asci, not at the tips of basidia that have 4 projectable spines upon which each of the spores are produced. So the false truffles have spores produced outside a special cell. True morel, or true hypogeous fungi or truffles have their spores borne in sausage-shaped tubes inside. So that's the basic difference. But they're gorgeous, and animals love them. They are edible, not terribly tasty to us, like the Gordian truffles, the white truffles and the black truffles that you find growing in association with oak and in Europe. We just don't have those in Alaska. But some of the roles that these fungi play, like this cup fungus, is actually growing on insect pupae. And so they love moths and butterfly pupae. And they will grow, not killing the insect until the propitious moment when the fungi determines it's time to make its spores. And then it just sucks all the goodies out of the insect, kills it, produces the spores, sheds its spores, and everybody dies. It's a terrible world out there. One shot, and you got to go for it. But look at some of these things, like this cordyceps growing out of these beetle larvae. And here's a grasshopper that has mushrooms growing from it. Another cricket, another little doomed organism. It's amazing what these arthropods put up with. And I'm sure you've heard about the ants that are infected with some of these things, do the dance, climb the trees until they can climb no further, lean back, attach themselves to the branch, and then out grows the fungus. And the fungus is able to release its spores up in the atmosphere to be carried on for infection of other animals. Mushrooms love mushrooms too. Some of the imperfect fungi, the hairy stuff with these little black specules that are sporangia, belong to the same group of mushrooms you think of as bread molds. Okay? And this bread mold, Spinellus fusiger, is actually growing on a gilled mushroom called Mycena. And here, have you ever heard of the lobster mushroom? Have any of you eaten the lobster mushroom? Ooh, we have one person back there who's eaten the lobster mushroom. I can't come to grapple with that and put it in my mouth knowing that one mushroom is decomposing the other. But that's exactly what's happening. The orange is a cup fungus, and it's devouring a gilled mushroom. So there you have a parasitic relationship. Hypomyces, and there are probably 4 or 5 genera of hypomyces here, something like that. Yeah. And they love the guild mushrooms in the family Lactariaceae, or the Russulaceae, excuse me. But they grow in us too, as I mentioned before. Toenail fungus, boy, some of them can get pretty bad. It's all curable though. There are azole chemicals, about 22 of them actually, that can be used to cure nail fungi very, very nicely. I've seen people coming back from the Vietnam War that had fungal infections on their back, skin infections, subcutaneous infections, and the chemical or drug ketoconazole would clear those things up in short order. It takes about a month, but they really clear them up quickly. And they grow and form what you might call eczema, ringworm, and thrush, crockrotch, or crotch rot, if you want to call it that, is another form of it. And I don't know how many people have seen ringworm, but it's very common in young kids, particularly households that have lots of cats, because cats oftentimes carry that particular fungus. And then we've got the lichenicolous fungi, and these are just the little black fruit bodies that grow actually on the lichen. So next time you pick up a lichen, you may actually be picking up two different organisms if you look carefully. And I mentioned the fact that the lichens were cup fungi, and here are the cups called apothecia that actually produce the spores of the lichen growing on another lichen. So it's just amazing what they do. And here's the one that was named after me, Polycoccum arsenii. It's not very pretty, is it? And then we have things like this that are very obvious, where all these little black spots are parasites on those lichens. Tim, I'm sure you've seen this stuff. And have you tried to identify any of the fungi? Yeah. So what biological roles do these fungi play? They're saprophytes, which means they eat dead stuff. They can be parasites, which means they're going to actually harm a host. They may not kill their host, but they're certainly going to degrade some portion of it. They can become mycorrhizal feeders, and about 99.9% of most of our plants are mycorrhizal, with the exception of some of the plants that have such finely divided root systems that they just don't need the fungal association. And these you'd find in some of these plants that are very common around here. And in plants that have their rooting system mired in anoxic muds. I mean, fungi are aerobic. They've got to have oxygen. So if the root systems of some of these plants grow in muds that smell like rotten eggs, you probably smelled some of this stuff before, you will not see mycorrhizal association. And of course, lichens, now a symbiotic association strategy with hosts that occupy fungi, yeast, algae, cyanobacteria, blue-green algae. And interestingly enough, Tim and others right here, were the people who discovered what the lichen association really is. So hand it to those boys for a wonderful job, well done. Mycorrhizae. What is a mycorrhizal association? Well, Greek myces is fungus, so I'm myces. You're rhiza. And I'd like to have your hand. Rhiza is a beautiful tree. And these are her beautiful rootlets buried nicely in the soil. She is emitting a substance called cyclic AMP, and as myces can smell it. I know that she's putting out a chemical that's attracting me to you. So Mikey's is going after rhiza. Root, Latin for root, Greek for mushroom. So the mushroom is now growing its hyphae. Oh, hold those roots up there. So that the hyphae come in contact with the rootlets and literally grow right around them and glove them. You're never going to forget this. And with that, we have what is called an ectomycorrhizal association. So now what I do for rhiza, I bring water and all the nutrients that are dissolved or soluble in water to you to feed you, and you have to feed me. Because up in your crown, you're absorbing sunlight and you're green. So what are you producing? Sugar. Sugar. So she's pumping sugar down to her rhizae because her roots are underground, but they still need feeding. So she's feeding her roots, but her roots are sloppy. And I'm so lucky that they're sloppy because I can then suck those carbohydrates up and I thrive on sugar. So you feed me and I feed you. So Mikey's and rhizae associate together, and that's what a mycorrhizal association is. Now we all know. So we study speciation using molecular genetics, micromorphological anatomy, and macromorphological anatomy. And Tim is a person who at UM is running the molecular genetics laboratory that sequences many of the things that biologists are trying to sequence. And so we're very fortunate to have him on board right here at UM because of the molecular genetic push in the speciation and delineation of species in the mushroom world. Well, fungal classification, as I said, is based on macro, and macro is what you see in terms of the fruit body. Micro, which you can only see with a microscope. And matter of fact, here is the outer epidermal layer of the root, and this stuff on the outside of the root is all ectomycorrhizal fungi. So there's the glove right there. And of course, molecularly, we're looking at a series of things, but primarily the internal transcribed spacer, or the ITS unit of that DNA to identify it. And the reason we use ITS, even though it may not be the best, is because that's what we started with, and we've got so much information gleaned from using, ITS, that we dare not forget it. So now we have to ask, how many of these are truly edible? Well, we found that about 5% of all mushrooms are truly edible. You can always eat every mushroom once. Okay. About 5% are deadly poisonous. So now you've got this gray zone. What's going on there? Some people will claim to have iron stomachs and can eat anything. I've known iron-stomached individuals, and mycologists too, who have succumbed to eating everything. So there are a lot of fungi out there that you can boil and actually pull the toxins out and get rid of them, like the false morels, Amanita muscaria. There's actually a recipe for fixing Amanita muscaria by cutting it up into small chunks, boiling it, throw the water out, boil it again, throw the water out, and then fry them up and then eat them. I wouldn't want to try it. And I don't encourage you to try that either. But some people go to great extent and lengths to eat some of these fungi. So many of these toxins, we just have no clue what they are. So where's the responsibility lie? Each and every one of us is in that responsible in that you need to properly identify your mushroom. You need people that have an experience with the mushrooms in your area and can adequately identify your mushrooms. If you don't know what it is or not sure, don't eat it. I have sat in hospitals holding people's hands who thought they were going to die from eating a mushroom that just so happened to have a toxin that was pulled out by their alcoholic drink that they had with their delicious steak and mushroom dinner. 18 hours later, they pull out and have wrenching pain from vomiting, but they live. But it's really hard to sit and watch somebody go through this agony. If you've ever eaten or never have eaten a mushroom before, and somebody feeds it to you as something being edible, be cautious. Eat only a small amount of it. I mean, less than a half of a teaspoon of it. A couple days later, if you're sure there's no reaction to it, you can go ahead and eat a little bit more. But day one, eat just a wee bit. Day two, you can eat another little bit to see if there's any reaction in you. Day three, skip. Don't eat day three because some of these toxins are bioaccumulative and you can get zapped on the third day. Wait for the fourth day, try it again, and see what might come up. So these toxins can be nasty things, and they're everywhere. And we'll be talking about some of these things later. Consumption of alcohol, people like to have a wonderful glass of wine or a pre-dinner drink, wine with the dinner, maybe even an aperitif afterward. And there are some fungi that are deliciously edible that if you have an alcoholic beverage, will extract a chemical that is ipecac-like in its nature. And you'll lose that dinner for sure. Big time. Gastrointestinal initiation of some of these toxins is pretty doggone common. And so not seriously, but can cause problems. So we come to the point where how do we begin to classify all of these things? There's so much variety in what we do. Well, we've used spores and spore colors to a great extent in our quick field identification, because you can oftentimes do a spore print in the field, get the color of the spore, and you can actually begin to place things into their groups or families by their spore color. And if you look at any field guide, almost all field guides start out with mushrooms that have white or lightly colored spores first, then those that have different colored spores, and I'll talk about them in just a moment. But you'll find fungi, even now that we're coming into spring, as you get your garden growing and your plants potted and ready to go, how many of you have seen little yellow mushrooms growing in your potted plants? Anyone? Guess where the potted soil's from? Georgia. Because these mushrooms, these beautiful mushrooms called Lepiota lutea are common growing. This is a picture from Georgia, as a matter of fact. But you'll find that mushroom growing in potted plants right here in Missoula, or we found in Fairbanks, Alaska. Other white-spored fungi grow on wood. Some grow as amorphous masses. Others have different structures from which they produce their spores, like gills or dangling teeth. And some of these are edible, some of them are not. Obviously, even though lenzites is an edible species, you just can't chew it enough. And it goes in mushroom and it comes out mushroom. And you don't want to take big bites because it can hurt. And then we've got those mushrooms that have pink spores, believe it or not. And some of these are edible. Like this guy is an edible mushroom, has pink spores, and gills that are free, but it grows on wood. It grows on wood. And so here we go, another pink spored mushroom. It's an edible. And here, Plutus nanus, another pink spored mushroom. And these tend to be edible. I've never eaten Plutus nanus, so I don't know if it is edible or not. But the pink spored mushrooms that have gills that are attached to the stalk often are poisonous. So you've got to know a little bit of the macro morphology of the mushroom to make sure you don't get into trouble. Free-gilled pink spores, edible for the most part. Pink spores, attached gills, don't even try it because they're just nasty things like the Entomomus species. Then we get into those spores that are really lightly colored, yellow-brown to kind of tannish, buff, dark buff white, like the Agrosipes and the Conosipes we see here. We'll see these on lawns in the fall. I'm sure you've seen them in your lawns in the fall. And these are poisonous mushrooms or will cause serious gastrointestinal upset. And this Bulbidius species, of which there are about 3 species, are actually edible, believe it or not. But they're so delicate and so small, it'd be hard to collect enough to even make a mouthful. So why waste your time when there are others that are huge? And this is what the Bulbidius looks like when young, and then the Agrosipes looks like this. And it grows on lawns, and these are decomposer mushrooms. Then we have the Agaricaceae, the ones that have brown spored fungi, and some of which are deliciously edible, like this very large Agaricus augustus is delicious. The meadow mushrooms you probably have picked. And if you want to see some of these mushrooms in the late summer, early fall, go to the golf courses, unless they spray their golf greens with chemicals. If you find mushrooms growing in your yard, and they are determined to be edible mushrooms, and you fertilize your lawn, don't eat the mushrooms. Because those toxins are absorbed by fungal tissue, and they're transmitted to you, and you don't want to get involved in that. But these are all edible species of chocolate brown spore groups. Then we've got the purple brown spore groups, like some of our dung fungi, and like the Psilocybe cubensis, the fungi that are referred to in the common vernacular as shrooms, that are sold for its hallucinogenic effect. Ironically, the chemistry of these things, psilocybin is the chemical that must be transformed in a mammalian body to psilocin, which is the inducement chemical that causes the hallucinations and or the psychedelic effects. So I've had to deal with lawyers, with folks who are being tried for having psychedelic mushrooms, i.e. mushrooms that have toxins, hallucinogenic toxins. And they've been able to argue their way out because the initial chemistry of the mushroom is not the chemical that induces the biological effect, desired effect that you see in mushroom eating. And we also see in our inky cap mushrooms, which are now black spored. So we go from white spored to faint yellow to cream, to yellow brown, to tan, to light yellow brown, light tannish brown, to light brown, to brown, to reddish brown, to purple brown, now to black. And these are the things a lot of people pick up and eat. You all recognize this guy in the middle? The shaggy mane? Yep. Deliciously edible mushroom, but they deliquesce, which means they produce an enzyme. It's like a cellulitic enzyme, and they will break down. And if you put them in your refrigerator, that enzyme is peaked to be active at temperatures of like 37, 38 degrees, which is exactly the temperature of your refrigerator. And the mushroom begins to deliquesce and turn to black oozy ink, and the whole mushroom dissolves and drips all over your stuff in your refrigerator. So, and you've probably seen them melt away in the forest floor too. Here's the mushroom that I've had association with people eating, having alcoholic beverages with, and getting terribly sick. Coprinus atramentarius. That's the one that produces that ipecac-like effect. So we talked about mycorrhizae being a fungus root association, but there are no less than 7 different classifications of mycorrhizal fungi as to what they do and where they do and with whom they do it. So we have endo or arbuscular mycorrhizae, a term that's probably no longer used, ectoendo, but it really refers to those fungi that start on the outside but will actually go inside, but do not necessarily compromise the cells of the outer lamella of the root system itself. But the ectomycorrhizae stay pretty much on the outside, but they also have cells that penetrate and grow down into the root itself between the cells. They don't grow into the cells. They grow between, and they go to the middle lamellae of the roots so they can deliver their power-packed nutrient bolus to feed those plants. And this is what some of them look like. You can see a little pine tree growing in its mycorrhizal association. And so we have different kinds of the mycorrhizae. I won't mention them here, but again, they may grow in association with the roots. They may grow into, but not penetrate the cortical cells, but they all tend to deliver their nutrients down here to the central stele if they're ectomycorrhizal forms. And this is what some of them look like. This is actually a root of a willow from Arctic slope. And I took one of these rootlets, cross-sectioned it, and the white stuff on the outside, that's all mushroom mycelial material. That's the glove right there. And in this photograph, you can see the glove out here. The glove actually stimulates the growth of the root system to have a particular shape or form. And this form right here is very, very typical of what you would find with an ectomycorrhizal association with a root. And some of these, like Tomentella, produce fruit bodies that are resupinate. In other words, they just literally form right along an underside of their substrate. So when you're out in the forest, roll over the log, and you're apt to see something like this, either that or lichens or other kinds of things. But again, it's this very thick mantle that we talk about when we actually talk about the ectomycorrhizal fungi, and the fungal hyphae then grow down between the cortical cells and into and between these other cells to finally get to the stele or the middle of the root to translocate those goodies upward. This is what it looks like under scanning electron micrograph. And you can see the mycorrhizae out here. You can see the root itself, the cortical cells, and the stele. And if you look at these hyphae carefully, some of them will have these bulges or buckles on them. And we call those clamp connections. And the clamp connections are only, well, one exception. One yeast actually has clamp connections. But for the most part, if it has clamp connections, it's going to be a mushroom. And some of those things that have white spores in the Hygropheraceae are slippery little suckers, but oh, they're so beautiful. And like Hygropheraceae conica, there's a whole slew of them that you folks will see here in our forest in the summer and early fall. We've got white spored and lightly colored to rosaceous, to yellow, to ochre spores that are produced in these russula species. But here's a russula that is actually an edible species. One of the 12 species of the green russulas, Russula virescens. And then Lactarius representaneus is a large, nasty old thing that just gets really slimy. A ectomycorrhizal former with spruce trees, primarily the white spruce in our country, but they're just gorgeous mushrooms. Then the pink spored guys in Enthalomas. The pink spored guys in the Paxillacea, actually it's a light pinkish brown. It's more of a clay brown, a dull brown spore color. However, this mushroom, Hygropheraceae, is a poisonous mushroom, gastrointestinally poisonous. It will make you sick if you try to eat it. But they're gorgeous little guys that grow oftentimes in our country, in Alaska, on cone scale middens. You know where squirrels go up in a tree and they start picking apart cones and eating the seeds, and they drop the scales down? Those cone scale middens are a great source for nutrients for some of these fungi. And then we come into this huge group of fungi called the Cortinaria. And a chap by the name of Joe Amirati and I are working on the quartz of Arctic slope. I've had Joe into the Arctic on several occasions. And my whole collection now of 23,900 collections all reside at the University of Washington under his tutelage, even though he's now retired, but they're in the Burke Museum there. But it's such a large group, 3,000 species. You'll find more quartz than anything else in our northern country. Ironically, no ectomycorrhizal fungi are found down south. They're all decomposer fungi, except maybe one. And you have to ask yourself the question, why is this? That's because those fungi are blown into those islands in the Southern Ocean from the southern tips of the continents. They come from thousands of miles away and land on those islands, and they germinate. If they're ectomycorrhizal, there's no trees, there's no host potential, and they don't make it. But they're beautiful fungi, but they can be really deadly poisonous. Foliotas, a lot of people eat these things. I'd be very cautious to have anyone eat them because they can cause gastrointestinal upset in lots of folks. But a lot of people do eat them. Then we've got the Gomphidiaceae, a group of mushrooms that has blackish, grayish black spores, and they're elongated. In fact, these guys are so closely related to the bolete mushrooms. Anyone know the mushroom, the steinpilz, the king bolete? Now we're coming to it. So those bolete mushrooms don't have gills. They have a spongial, a porous spore-producing hymenial mechanism underneath the cap. And these guys are so closely related to them that they now are placed genetically very close. But then you've got the beautiful Amanitas, again, white spores. And this is not Amanita muscaria. It's one of the most common Amanitas that you're going to see around here, Amanita muscaria variety flava vulvata. And that's the mushroom that has caused most of the problems with gastrointestinal upset and poisoning of people. Wherever I've been in the world, I'll get phone calls from poison centers asking me to identify mushrooms that either dogs eat, little kids eat, or grown-ups who know better but still eat. And one group of young people, age range from 18 to 28, out of 12, 11 of them ended up in the hospital, and they all ate this particular mushroom because it has 3 toxins, one of which is an hallucinogenic toxin. So they're out there for the kicks, but boy, did they get kicked. And there's some beautiful audible sounds that are now being recorded that mushrooms make. I had one here, but it doesn't work with this system. But you can go to any one of those YouTube URLs and just simply type in mushroom music, and then open up one of the YouTubes, and the mushrooms literally sing. It's just amazing, the noises that mushrooms make in the forest amongst the forest floor critters. Well, we have been working primarily, again, in the circumpolar Arctic. So I've been in a good portion of the Russian Arctic and the US Arctic. I've not spent a lot of time in the Canadian Arctic. And I've spent time in the European Arctic as well. And the subarctic, and then island archipelago. So the one that you would recognize being the Aleutian Islands is very similar to those that we find in the Southern Ocean. Those islands look very much like the islands you would see in Alaska. In the subantarctic, here's Antarctica. And here is our southern tip of South America. We have the southern tip of Australasia, in other words, New Zealand and Australia and Tasmania. And over here, we've got Africa. And the Southern Ocean has an interesting line, being 2 lines, the Antarctic Circle. But this July 10 isotherm, and below which there's ice prevalent and glaciers, above which there are none. So in working in the Australian islands, Heard Island is actually below this line, but Macquarie Island is above this line. And the 2 islands are phenomenally different. I mean, one has glaciers on it, the other one does not. And if invited back, I'll give you a presentation on subantarctic fungi from some of those beautiful islands down in the Southern Ocean. But we were really concerned about the land bridges and where these fungi came from. And this is the actual extent hypothesized of what the Bering Land Bridge was about 46,000 to 24,000 years ago. And so you have a vast connection between what is now Alaska and what is now Russia, where migrants, not only people and animals, but mushrooms actually were able to be transported back and forth by wind-blown spores. And it's only in this region right here that we see all 7 species of Amanita muscaria. I'll show them to you. Amanitas are native to the Northern Hemisphere. They're genetic and morphologically varied. All 7 varieties are widespread throughout Alaska, and this genus is widely spread throughout the world, but not all 7 are found anywhere else other than Alaska. They're commonly found in boreal forests. They're all ectomycorrhizal, and we have color variants like Amanita muscaria variety alba, pure white, but it is Amanita muscaria. And then we have Ammonite muscaria variety Formosa, which is a straw-colored yellowish tan mushroom. Gorgeous. And then Ammonite muscaria variety Persicina, sometimes referred to as Gasawii, but it's the same thing, actually, we think, that has a peach-colored cap. And then we've got Ammonite muscaria variety Flavovulvata that has the red cap that goes to yellow and to white around the margins, and is the one that causes most people most problems because it's the most plentiful of the Ammonites that I've seen, at least in North America. Then we have Ammonite muscaria variety muscaria. Note the difference, the pure red cap and the white squamules on top. So typical of that particular mushroom. Is it any wonder that in northern Scandinavian countries, where this mushroom was very abundant, that Santa Claus has a red suit with white trim? There is a relationship. Ammonite muscaria variety Regalis is a brown cap thing. And here you can begin to see the composite of these things found only in Alaska altogether. And it was a wonderful find. Joseph Gemmel, who's now in Hungary and doing work there mycologically. Lee Taylor, who was at the University of Alaska, is now at the University of New Mexico. The three of us pulled this story together, and it's a fascinating story because Ammonite muscaria is such a common mushroom, but again, with the 7 varieties only being found in one state. So we have in continental separations, we have groups we call clades, but groups of mushrooms, group 1, group 2, and group 3. Group 1 being found primarily in Europe and Africa. Group 2 being found in Australasia. That means New Zealand, Australia, Tasmania, etc. And group 3 in North and South America. Then we've got Alaska, which is the hot spot, and we have forests, humid forests in Northern Asia and Alaska are inferred as the center of the origin of this particular mushroom. And I point this out because this mushroom is so common to so many of us. And we see it growing here, and they're just so beautiful when you come across them. And ancestral populations probably were subdivided by the opening of the Bering Strait about 12 million years ago, when things became rather aquatic and they could no longer travel or be transported easily. Cooling climates, no northward migration being observed. Beringia remain the continuous habitat for Ammonite muscaria, and their morphological varieties are now suggesting that they should be separated into their own species and not just as varieties. And that's probably what's going to happen, and the genetics will drive that. So now, there's this whole other group of mushrooms out there. Mushrooms that don't have gills. So what are they? They're mushrooms that have blunt gill-like ridges, or pores or teeth, or sacs, or are gelatinous. They have pored undersurfaces, or coral-like teeth, earth tongues, cups, bird nest fungi, rusts and smuts and tar spots and powdery mildews, imperfect asexual fungi. Boy, they're everywhere. Matter of fact, I got an experiment for you. You think when you talk to your mother or your wife and she says she has a clean kitchen, take a piece of homemade bread and rub that bread softly across her countertop. Turn the bread over. Take a cottage cheese container, clean. Put some paper toweling in the bottom of it, moistened, but don't get it sloppy wet. Put the piece of bread on top of the moistened paper towel. Put saran wrap on the top of the cottage cheese container. put a rubber band around it, because what's going to grow there, you do not want it to get out. And then you can show your spouse, you need to clean a little better. And so what are some of these fungi? We find the Gomphus group that have the blunt ridges. They're not gills, blunt ridges. Do you recognize this one? The golden chanterelle, a delicious edible, you bet. And here it is again. Then we've got those that have pores underneath or gilled structure underneath. This guy actually has gill-like lamellae on the underside of this bracket unit. This guy has pores. Here, this guy has pores. This guy has pores. Here are pored surfaces. This guy has a pored surface underneath. And this Ganoderma apollinatum is called the artist conch. Are you familiar with it? Have you drawn pictures on it? Oh, you do. I have seen some beautiful etchings. The hymeneum surface is pure white when fresh. And then what happens, you can etch the surface, and the surface will dry, and where you've etched remains dark. And you've got the fingerprints all over it. Yeah. And so, again, the pored fungi. Here are some teeth fungi. Very common in this area, I'm sure. There should be 5 or 6 species of this particular sarcodon in our country, but here are the teeth. And Hydnellum, and the Hericium species. Lion's mane, does that ring a bell? There you go. And Hericium coralloides, another very delicious edible, oftentimes growing on birch. Then you got the puffballs, right? And those puffballs can be pretty doggone large, like Calvacia booniana. This is the largest puffball I have ever seen in Alaska, growing at 3,200 feet in Denali National Park. And then Bovista, again, a small puffball, but tends to be kind of moist on the inside when fresh. But Lycoperdon are edible if you cut them in half. And if there are any rules of thumb in mycology, this is probably one of the best. If you cut a puffball in half, and it's truly a puffball and not an immature mushroom, and it's white on the inside, chances are it's probably edible. I have seen people pick up that exact mushroom, and I took a bite out of it and chewed it, and then the rest of the class did the same. One lady put it to her lips, and she said, Dr. L, and her tongue started swelling, her lips started swelling, and she was having an anaphylactic shock to what I would consider to be a very edible mushroom. That's why you want to be careful. I've never done that since in my classes. I don't fry mushrooms and give them to my students, because I've seen people from Europe where there's a mushroom called Collibia dryophila that is sold in the marketplaces in Europe. And in our country, Collibia dryophila, 2 mycologists from Michigan, and not the University of Michigan State, had a class out. They found this mushroom. Everyone in the class ate the mushroom except the TA and the professor. Everybody got sick except the professor and the TA. That's why I don't do that anymore in my classes. It's just too risky. Then you've got the earth stars. Oh, they're beautiful. And they come up, mycologists come up with some phenomenal names for some of these things, like Gyastrum fornicatum. Where did they get that? I don't know. But you never know. And the jelly fungi. Feel your earlobe. Right now, just touch your earlobe. You've just touched a jelly fungus like, and feel, okay? And the boletes, those that have pored surfaces underneath. And here's the king bolete over here and being held by this little Buddha. Deliciously edible fungus. I've never known anyone to get sick from this. If anyone, any of you know someone who gets sick eating this, I'd be liking to know that. And the coral fungi, many of which are edible, but many cause gastrointestinal upset, particularly those in this particular group of mushrooms in the Romeria genus. They tend to be gastrointestinally upsetting for lots of folks. And then the earth tongues in Geoglossum and Spathularia. They look like little tongues running right out of the mouses. And then the cup fungi. And these otidias are called pig's ears because it looks like a pig's ear just kind of growing right out of the ground. Beautiful. And the Microstoma protacta is a cute little guy, and it fruits early in the spring. So if you get out in the woods and you find a bed of scoriaceous plants or equisetum plants, chances are you will find this fungus because they tend to be associated with the rhizinae, or the underground root system of our scoriaceous plants. And the bird nest fungi. They look like little nests with eggs in them. And the rusts. And it's the rust that causes the witch's broom we talked about earlier on. And the smuts, growing on a lot of our grass species, maize. This particular smut is eaten abundantly in Mexico. Have any of you ever eaten corn smut? Eustalaco maize? Ah, was it good? Interesting. I've never eaten it, never had a chance to eat it. But when it's mature, it's powdery dry. And of course, you wouldn't be able to even choke it down. Then we have tar spots. Again, cup fungi, ascomycete. We've got the Eryciphe plants that grow on many people's garden plants, particularly the cucurbits, the squash, those plants that have really big leaves, a pumpkin, and other things as well. And the uncinnulas are typically found on, at least in our country, on the willows. But those are each of those little black spots is an individual fruit body. So that leaf is just chock-a-block full of fruit bodies. And of course, then you've got those molds that really go after your fruit if you let it sit too long. And the penicilliums are very abundant, well over 120 species. And in 1917, there was a gentleman who was working with cultures and a bacterium and bacterial infection. And on the windowsill of his laboratory, he placed these dishes, and he noticed that in some of the dishes, the bacteria were being killed. And he wasn't sure what was going on. And come to find out, the dishes were contaminated with one species of this particular mold, Penicillium notatum. And from that, we have found that some Penicillium species produce these droplets which contain penicillin. So Penicillium produces penicillin. And it was during WW2 that this was really brought to the forefront, and that much of the initial experimentation with penicillin was used by many of the fellows that were coming back from the Pacific theater who had fungal infections, and penicillin just nailed them. And of course, we see all kinds of variants now of penicillin-like materials. And then, of course, the slime molds I mentioned earlier, they're beautiful, but again, no longer are they in the fungal kingdom. So we ask, what makes a useful specimen? Well, you got to collect the whole thing. In other words, you got to get down into the soil in some cases and make sure you get the bottom of it. You got to have a place to store them so they don't decompose. And that does not mean placing it in a plastic sealable bag. Paper bags are the best. I use those sandwich bags, wax text sandwich bags for collecting. They work beautifully. You got to make a note of the substrate. Where did you actually find it? What was it growing close to? And then you got to transport it back to your laboratory place for study. You got to label them. You should photograph them. And you should preserve under very low moving heated air. Don't put them in your oven and try to heat them and preserve because you'll bake them. And then, of course, you want to accession those into your collection. And of course, describing them using fungal keys and field guides will really help you out. This is a very dear friend of mine from Germany, who is a bolete specialist. I've had in Alaska several occasions, and looking at some of these plants. And here you can see the collection of the fungi, laying them out on a dryer shelf, labeling the mushrooms, describing, photographing, using color keys and codes, describing, and then place it onto a heating shelf. And with catalytic heaters, had slow moving, warm air moving up through. And that's how we preserve fungi forever and ever and ever. Just dry them out. So fungal determinations are based on spore color, fruit body color, fruit body taste. Yes, I taste everything. But I don't swallow. Like Bill Clinton said, he didn't inhale. Okay. Staining reactions, really important to note. The roles they play in the natural setting. Their aesthetics. I mean, they're beautiful to photograph. And they're in some cases delicious to eat. They're diverse and their shapes are phenomenal. Commercial use, we use fungi all the time in fermentation, cheese making, and cheese aiding. If you go to the grocery store and look at packaged, dried product, you'll be amazed at how many of them actually have fungi in those packaged products. I went to a Safeway store and I found 12 different items that had fungi in them. And if you're allergic to fungi, it could be a real problematic situation. And of course, for commercial spills, and I've had students working with the fungi that will actually decompose organics like oil, grease, gases, and whatnot. They, like I said, fungi live on anything, and they love tar. I've seen where in Cape Simpson, the oil has bubbled up out of the ground for millennia, and it's formed rivulets and formed a large wedge to the sea of moving oil that when it loses all of its hydrocarbon evaporative materials, just turns to tar. And I've seen mushrooms popping up through all of this stuff. It's amazing what they're able to do and what they're able to live on. All they need is the carbon source. And oil and tar is rich in carbon. And yes, some of them actually glow in the dark. And that's it for tonight.